From the insulin that keeps a diabetic healthy to the enzymes that get your laundry clean, our world really runs on these tiny, microscopic factories. But who are these little workers? Well, today, using Clark and Pezdernik's biotechnology as our guide, we're going to meet the powerful and honestly the unsung workhorses of the biotech world. All right, so here's our game plan. We'll kick things off by asking why the lab couldn't just stick with one superstar organism. Then we'll get into plasmids. Think of them as the genetic software that runs the entire show. From there, we'll look at a massive industrial problem, take a trip into the incredible kingdom of fungi, and then we'll wrap it all up by learning how to pick the perfect microscopic tool for any job.
So let's set the stage here. For a long time, molecular biology had a clear MVP, a go-to organism that did almost everything. But as the field got more ambitious, so did the challenges.
So you might be asking, why bother, right? Why couldn't scientists just stick with the one tool they knew inside and out? Well, the simple answer is their ambitions got way bigger. The problems they wanted to solve became way too complex for a one-size-fits-all approach.
For years, that superstar was E. coli. And for good reason. It's pretty simple, it grows incredibly fast, and we understand its genetics really well. It was, and honestly it still is, the perfect model for figuring out the basic rules of DNA and proteins.
But here's the thing, E. coli has its limits. It's kind of like having a brilliant workshop that's perfect for building one-off prototypes, but you'd never use it to run a massive global assembly line. For those huge, complex jobs, like manufacturing human proteins, scientists realized they didn't just need one tool. They needed a whole new toolbox, and it had to be filled with specialists.
Okay, so if these different organisms are the hardware, what's the software that tells them what to do? That brings us to the universal operating system of genetic engineering, plasmids.
So what exactly are these things? Well, in a nutshell, plasmids are these small, independent rings of DNA just floating around inside the cell. They're totally separate from the main chromosome and, this is the crucial part, they can make copies of themselves on their own. Honestly, the best way to think about a plasmid is like a tiny programmable app. You can load it up with new genetic code, pop it into a cell, and get that cell to run a totally new program, like, say, producing insulin, or even glowing in the dark.
So what makes these little apps so incredibly powerful? It really comes down to a few key features. They exist outside the cell's main DNA, they can copy themselves, and you can load them up with all sorts of new genetic instructions. It's no wonder the source material calls them the crux of all molecular biology. They really are.
Okay, let's see this stuff in action. We've got our software, the plasmids, but now we need the right hardware for a huge industrial job, getting valuable proteins out of the cell factory and into our hands. This is a massive bottleneck. I mean, think about it. You can engineer a cell to produce tons of a useful enzyme, but if that enzyme is trapped inside the cell, it's really difficult and expensive to purify. What you need is a cell that's great at exporting its own products. And the solution, believe it or not, lies in the cell's basic architecture. E. coli is what's called gram-negative, which means it has two cell membranes, kind of like a double-wall security system. Getting a protein out is this complex, multi-step headache. But a gram-positive bacterium, like Bacillus subtilis, has only a single membrane. It's like having just one simple door to walk through, making it a natural-born expert at shipping its products out. And just like that, by choosing a different workhorse, the entire problem is solved. Bacillus subtilis is a natural exporter, perfectly designed for just churning out massive quantities of enzymes for everything from laundry detergents to making our food.
So far we've been hanging out in the world of bacteria, but what if we need to study cells that are structured more like our own cells? For that, we need to jump into an entirely different kingdom of life, fungi.
Think of it this way. Bacteria, or prokaryotes, are like simple studio apartments. Everything is just in one big room. But our cells, eukaryotes, are like complex houses with lots of different rooms for different jobs, like a nucleus for the DNA. To study our own complex biology, we need a model that has that same house-like structure, but is still simple enough to work with in the lab.
What's wild is that humans have been using fungi for thousands of years, long before we even knew what they were. They're the original biotechnologists, the little critters responsible for fermenting our beer, making our bread rise, and creating those amazing flavors in cheese and soy sauce. And they aren't just tiny chefs, they're also miniature chemical factories. The most famous example, of course, is penicillin, the life-saving antibiotic that was first found in the penicillium mold. And on a huge industrial scale,
Other fungi are used to produce chemicals like the citric acid you find in so many foods and drinks.
And in the lab, one fungus is king, Saccharomyces cerevisiae, which you probably know better as baker's or brewer's yeast. It has become the absolute go-to model organism for studying the fundamental biology of complex eukaryotic cells.
The magic of yeast is that it gives us the absolute best of both worlds. It has the complex internal machinery of a human cell, a nucleus, chromosomes, the whole shebang, but it's still a simple little microbe that grows at lightning speed. And the real kicker? It has its own version of a plasmid, which makes it just as easy to genetically program as any bacterium. It's a total game changer for research.
So we've got bacteria for speed and simplicity, we've got industrial specialists for large-scale production, and we've got yeast for studying complex cells. This brings us to the final and most crucial point of all.
This is really the essential takeaway. The power of modern biotechnology isn't in finding one singular best organism. Not at all. It's in understanding the unique strengths of a diverse cast of characters and then choosing the right one for the specific task you have at hand.
So the modern biotech toolkit is all about specialization. If you need to copy and move genes around, you use a plasmid. That's your mobile genetic software. For a huge enzyme factory, you call on Bacillus. And to explore the inner workings of a complex cell like ours, yeast is your go-to model. It's always about matching the right tool to the right task.
And this quote from the source really just hits the nail on the head. The entire revolution is built on this one ability: to take a gene from one organism, a so-called heterologous protein, and get it to work in another one. That simple concept is what unlocks all of this incredible power.
So we'll end with this thought. For all the amazing workhorses we know and use, scientists estimate that we've only successfully managed to grow a tiny fraction of the microbes here on Earth. This leaves us with a pretty fascinating question. What specialist, with what unique new ability, is still out there just waiting to be discovered?